Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, my name is Abdul Gafur. I'm from the Faculty of Mathematics and Natural Sciences, Universitas Lambung Mangkurat in South Kalimantan. Today, in this presentation, I am going to talk about an identification key to orders and families of soil and freshwater nematodes. For those of you who are not familiar with uh, soil nematodes, I can briefly say that nematodes are microscopic worms that are one of the key components of soil biota in soil ecosystems. They have the highest diversity, the highest number of individuals, and also the highest biomass amount among soil animals or soil metazoans. And ecologically, they occupy different trophic levels in soil food webs. As you can see in this picture, they appear in the second trophic level as root feeders or plant feeders, in the third trophic level as fungal and bacterial feeders, and also in the fourth trophic level as predatory nematodes. Because nematodes are the main components of soil biodiversity, they have the potential use in assessment of soil general biodiversity. Because they are important components of soil ecosystems, to some extent, nematodes can determine and reflect soil conditions. So they have the potential as bioindicator of soil condition or soil health. But in order to achieve the two potentials, we need identification skill. So workers or students have to identify their nematodes, and that's a problem for us in Indonesia, at least because of the microscopic size of the the microscopic size of the nematodes. um, we need to have a good microscope, of course, and also a good skill in microscopic examination of nematodes, because the nematode body are soft. They decompose very quickly once they die. So we need to have a good skill too in preparation of specimens of nematode for microscopic examination. And the third is the lacking of identification guide or identification key of soil nematodes, because we haven't studied our nematodes sufficiently. And last but not least, we are lacking or facing a declining number of experts who can identify soil nematodes. In this presentation, I'm focusing on the lacking of identification guide or identification key of soil nematodes. So, to fulfill the urgent need for identification key of soil and freshwater nematodes, I have developed a dichotomous identification key that help in identifying nematodes to order and family levels. It is equipped with many pictures in almost every step, so it is a pictorial key, and also it is written in Bahasa Indonesia because it is meant to be used by Indonesian workers. In developing the key, I followed the general classification of nematodes by Delay and Blackster, and then for the more detailed classification, I followed classification or scheme by other authors. So now the key covers 14 orders and 23 families of soil and freshwater nematodes. Not all order and family of nematodes covered because this key is only for use for identifying uh, soil and freshwater nematodes. And in developing the key, I decided to make separate keys instead of one all-inclusive key. So there is a key for orders, one key for orders of nematodes, and then followed by separate keys to families of each order. Now I would like to show you a short video of the key which is available online free for free in this website. So this is the key, um, and as I told you, this key is equipped with extensive number of pictures, so it's a pictorial key, and this one key is for orders, for orders of nematodes. And then once the order have been identified, we can follow the families by clicking the order of the nematodes. For example, here order Aphelenchida will lead us to a separate key for families of this order. If the family consists of only one genus found so far in soil or in freshwater nematodes, then the genus will be written here as well. So at the same time, this key is not only to family but also to the genus of the nematodes. For future improvement, I plan to add more photographs of real specimens because workers use real specimens, examine real specimens. It will be helpful for them if they compare with photograph of real specimens too. And then it will be helpful if I add images of specimen from Indonesia, but that means that I need more nematodes collected from Indonesia too. And I also plan to develop a multi-entry key instead of only a dichotomous key, because this multi-entry key is easier to use, more user-friendly, and also it's easier to revise when the taxonomy of the nematodes change. As a summary, I can say that I have developed a pictorial dichotomous key to orders and families of soil and freshwater nematodes. It is freely available online, and hopefully this key will encourage and facilitate the use of nematodes as bioindicator in biomonitoring of soil and freshwater health in Indonesia, and in turn it will help in maintaining the soil and freshwater health in Indonesia. That's all for my presentation today. Thank you very much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.